Hello to all the researchers, practitioners and all people who are part of the GBike network, with whom I'm constantly in contact. For me, it's a crucial point of reference for obtaining scientific advice, sometimes even in frantic conditions, during these 3 years of scientific and negotiating process that we hope will lead to an ambitious, inclusive and transparent global agreement to halt and reverse what some are calling the biological annihilation of the planet. As you know, in December 2022, the 15th session of the Conference of the Parties to the Convention on Biological Diversity will decide on the so-called Post-2020 Global Biodiversity Framework and set new conservation targets for 2030 and goals for 2050 for the conservation of biodiversity, while ensuring sustainable development and benefit sharing. Genetic diversity, together with species and ecosystem diversity, is recognized as one of the 3 building blocks of biodiversity under the United Nations Convention on Biological Diversity. Still, the Convention has among its 3 objectives, in addition to conservation and sustainable use of biodiversity, also that of access to genetic resources and the fair and equitable sharing of the benefits deriving from their use. You have made clear the reasons for protecting genetic diversity. Inter alia, it is the basis for evolutionary change, and it is critical that species adapt to climate change and biotic interactions, including new pests and diseases. Indeed, there is abundant evidence of the substantial role of genetic diversity in the resilience of ecosystems and in the conservation of species diversity. Genetic diversity helps maintain ecosystem functions, stability and services. Besides, the use of limited genetic stocks in forestry, fisheries, horticulture and agriculture can result in large social, economic and cultural losses. As the most significant global conservation policy mechanism, the new CBD framework has far-reaching consequences, and it will guide conservation strategies and actions and subsequent reporting for each CBD member country until 2050. In previous CBD strategies, goals, targets and indicators for genetic diversity were undeveloped and mainly focused on a few major agricultural species. Yet we are aware that to achieve global conservation goals, genetic diversity within all species, not just domesticated species and their wild relatives, must be conserved and monitored using appropriate metrics. Basing ourselves on the suggestions of this scientific community, and more generally on the fruitful collaboration between you and the group of experts which provides support to the Italian Ministry of the Environment and Energy Security, we have extended the scientific acquisitions and the argumentation for 3 new pragmatic indicators for genetic diversity and modifications to the 2 current indicators for maintaining genetic diversity and adaptive capacity of all species. The indicators are the number of populations with effective population size greater than 500, the percentage of populations maintained within the species, and third, the number of species and population in which genetic diversity is monitored with DNA-based methods. These targets and indicators, which we consider pragmatic, may have uses behind the CBD commitments. In fact, they can be extended to measure progress towards the implementation of similar targets developed within European, national and local policies. For example, to measure the achievement of conservation objectives in protected areas or in sites identified as valid for biodiversity protection or restoration. With this, I finished my intervention. I would like to thank you again for inviting me, and yeah, goodbye.